Welcome to Horn. This is a lecture for the teaching modules of the One Health Regional Network Horn project. This lecture forms part of the Tier 2 Generic and Transferable Skills Curriculum programme. My name is Marie McIntyre and I work on the epidemiology of human and animal disease at the University of Liverpool in the United Kingdom. The subject of this lecture is critical appraisal of quantitative research. Quantitative research occurs in many different fields, for example, sociology, anthropology, economics, ecology, infection biology, epidemiology, and climatology. As a result, it is difficult to provide a specific list of questions to ask when undertaking a critical appraisal which will cover all of these topics. However, generally, when examining some research, you can gain an overview of the quality of the results by asking certain questions. The answers to these indicate how thorough the researchers have been. For example, is the research question original? Is the research question important? How does the research fit with other work in the field? Does it say something different? If so, what and why? Were appropriate methods used? Can you see any problems with them? How much effort went into developing the study? Further questions include, are the results credible? How strong was the evidence for the findings? Are the conclusions consistent with the findings? Do the findings add to the research field? What did the author say about other literature in the field? Is the paper clearly written? Does all the content seem logical? Where was the author from? Do they have any obvious preconceptions about the subject? What is their academic discipline? Quantitative and particularly epidemiological studies can use specific criteria to assess research. These describe the evidence in the study, the internal validity of the study, the external validity of the study, and whether the results of the study are comparable with those of other studies. The description of research evidence is examined by answering a series of questions. What was the main aim, the hypothesis of the study? This should be clearly stated. A study should be designed to address a particular research question. Sometimes several similar questions can be addressed simultaneously, but well-designed studies are rarely able to address many questions simultaneously. What was the main study factor? In epidemiological studies, this is likely to be the exposure or intervention. In some studies, the study factor will be obvious, and it's often stated in the title. However, in other work, this may be less clear, or the study may be exploratory, investigating many potential study factors. Studies investigating many study factors are more likely to detect statistically significant findings by chance, creating type 1 errors. What was the outcome factor? This should be clearly stated. Some studies will have more than one outcome of interest. Multiple outcomes may lead to multiple statistical tests being performed, which increases the risk of type 1 errors. What was the study design? It is not uncommon for the authors to forget to state the study design or to get this wrong. The most common mistake is to declare a case-controlled study has been performed, usually when cases of a disease are involved, when really a cross-sectional or cohort study design was used. Cohort and intervention studies demonstrate causality, whereas case control and cross-sectional only demonstrate associations, which may have occurred by chance or due to biases in the data. The triangular diagram describes a pyramid of the quality of evidence when studies are well run. Study designs with the most rigorous evidence include, when applicable, randomized controlled trials, RCTs, and systematic reviews of these. Further questions include, what was the study population? Can you define the study population in terms of species, their demographic characteristics such as age, sex, ethnicity, or breed, the location of the study, where it was undertaken, and whether this was representative of the population the study was trying to examine, or are the results likely to be biased by the research population and location, And time. Could changes in some factor over time impact the study results? What was the main result or results, and is the author describing these? This should relate to the main study aim. However, some authors often discuss the most statistically significant results or even the most surprising results. Sometimes results for the stated main hypothesis are ignored altogether. Remember, when many results are presented, there is an increased risk of type 1 errors. It is safest to concentrate on results relating to the main hypothesis, particularly when that hypothesis was generated beforehand. Ensure that the results really are what the authors say they are. For example, do they confuse incidence and prevalence? Ensure that such calculations use the most appropriate numerator and denominator, and also ensure that the conclusions can actually be made based on the data collected. The internal validity of a study is the degree to which it represents the true situation in the study population. When assessing internal validity, consider: is selection bias likely? Could other factors confound study results? How was the study population selected? How many individuals were available for analysis? Convenient samples are likely to be biased. If you cannot determine how a sample was selected, assume the worst. Be cautious when authors say a sample was randomly selected. This might mean I wasn't conscious of having selected particular individuals, or that no true form of random selection was used, for example based on random number generators or random number tables. Consider the number of subjects lost during studies and before analysis. This may be due to non-participation of study subjects, or because some data was not collected for subjects. The more individuals which are lost, the greater for the potential for biases of results. Why was data lost? Is it due to bad study design? Did authors compare, using available information, subjects lost to follow-up and those included in the analysis? If data for these two groups are not comparable, we should have little confidence that they will be similar in other ways. The effect that missing data has on study results is hard to determine. It is a function of the number of subjects lost and the degree to which those lost differ compared to those remaining. It is not always possible to determine from the information presented how many subjects were lost. If this is available, did the authors examine remaining information for biases? It can be informative to calculate best and worst case scenarios. For example, estimate the effect, the odds ratio or confidence limits, for example, if all missing subjects had the exposure or outcome of interest, or vice versa. If the results under each scenario lead to the same general conclusions, then selection bias due to loss to follow-up will not substantially alter study conclusions. Remember that some study types are particularly prone to certain types of selection bias. For cohort studies, determine if the exposed and non-exposed animals are from the same population. For case-controlled studies, determine if the cases and controls are representative of the same population. Are observation and other measurement biases present? Do all the measurements used conform to standards deemed necessary to obtain true values? Often it is difficult to measure the exposure we are actually interested in, so we measure a de facto or proxy factor that we hope will tell us what we need to know. Is this measurement appropriate? Was action taken to avoid differential misclassification? For example, if more than one instrument or observer was used to make measurements, were these assigned equivalently or randomly to the groups being compared? Is confounding likely to be present? Once the exposure and outcome factors have been identified, you should make a comprehensive list of possible confounders. Clearly, this requires knowledge of the subject. The paper should be examined to see how the authors have dealt with these confounders, if at all. Potential confounding may be dealt with during the design of the study or during the analysis. At the study design stage, were these dealt with using restriction, matching in a case-controlled study does not control confounding unless analysis is done taking account of matching, and randomization. This is the only method able to prevent confounding by factors which have not been considered in the study. At the analysis stage, Was confound and controlled within analysis by demonstrated comparability? For example, the potential confounder is equally distributed amongst the exposed and non-exposed groups in a randomized trial or cohort study, or amongst the exposed and non-exposed controls in a case-controlled study. Analyzing data in strata of the confounder, for example, age-specific groups, or adjusting the confounder using a variety of statistical techniques, for example, multiple linear or logistic regression modeling. Are there issues with the statistical analyses presented? Is there sufficient power? Low statistical power increases the risk of type 2 errors, i.e. failing to find a significant result when really the outcomes are different for the study groups. Low power is a substantial problem in medical and veterinary research. Even when a study has sufficient power, power is reduced if data are analysed in strata, so-called subgroups, as clearly each of these strata will have less subjects than the overall analysis, and if some individuals are lost to follow-up. When multivariable regression has been conducted, it is important to remember that only individuals with data for all the variables in the analysis are included. So if data for one of the variables is missing for many of the subjects, then these will be absent from the analysis, resulting in a reduction in power. If the researchers don't check for this inadvertent loss of subjects from the analysis, they may not even know it has happened. Are appropriate statistical methods used? It is unfortunate that many studies use inappropriate statistical tests. Common examples include the use of parametric tests when data is not appropriately distributed. The summary data presented in a paper can sometimes be used as a quick guide as to whether the data is normally distributed. In normally distributed data, the mean and median are similar. Also, 95% of the data will lie in the range of 2 standard deviations above and below the mean value. If the lower value of this range lies below 0 for a variable that clearly cannot truly be negative, such as body weight or age, then the data is likely to be non-normally distributed. Another common problem is multiple statistical testing. As mentioned elsewhere, increasing the number of tests performed increases the chance of finding a significant result just by chance. A simple and conservative way to assess the potential effect of this is to use the Bonferroni correction. This simply reduces the critical p-value needed to declare a result significant. It is not unusual for statistical methods to be poorly described, applied, and interpreted. For example, a temporal association between exposure and outcome is only possible with cohort and intervention studies. It's not possible with case control and cross-sectional studies. And within this temporal association, remember to see that if there's a dose-dependent effect. Finally, if a statistically significant relationship has been found, does this actually mean something? Is the relationship both strong and important? External validity means whether study results are applicable to other populations, i.e. in the real world. Laboratory studies can have excellent internal validity but lack real-world application. Disease prevalence studies may not be applicable to other geographical areas or time periods. Restricted studies may not be applicable to all groups, and it's important to consider what the study population represents. Study external validity only occurs with internal validity. Generally, the authors will use the discussion section of a paper to put the results in context with other published information. However, this discussion is often not exhaustive and authors may concentrate on results that support their findings. Hence, it is important to consider evidence in addition to that provided in the discussion section. It is necessary to determine if the results are consistent with evidence from the same and other species. Furthermore, it is possible that authors have misinterpreted or misquoted the results of other studies. A simple rephrasing of a sentence can dramatically alter the interpretation of a statement, and the effect may be not obvious. Therefore, it's important that references used by authors are checked, particularly when they are important to the author's conclusions. Check the authors. Do they have any obvious preconceptions about the subject? What is their academic discipline? This impacts their approach. It is also important to decide whether the results are biologically plausible. Readers should be cautious about believing results that are difficult to explain. Similarly, authors may focus their discussion on findings that were unexpected or surprising. Care should be taken when interpreting such findings, particularly if they were peripheral to the main aim of the study, as they may represent type 1 errors. It is important to consider who the authors are and who funded the research, as there may be a potential conflict of interest. However, readers should not be too cynical. Finally, it is worth mentioning the phenomenon of publication bias. This is where studies that find negative results are less likely to be published. That is, studies that do not find a significant effect of an exposure are less likely to appear in the peer-reviewed literature. This has at least two potential effects. Firstly, researchers may identify a single study suggesting that a certain exposure increases the risk of a disease, whereas numerous studies that fail to find such an association were not published. Secondly, scientists may search for significant results in order to publish papers about studies they spent a lot of time and effort conducting. So a study which was designed to investigate the effect of one exposure on disease X may be used to write a research paper describing the effect of a different exposure on a different outcome if these factors happen to be measured and turned into a statistically significant relationship which could be mentioned within the study. This approach greatly increases the risk of type 1 errors, but may be difficult to detect when a paper is reviewed. Please now view the lecture on appraisal of qualitative research.